This card displays a microbial community called the Winogradsky column. The Winogradsky column is a microcosm of how bacteria interact on the surface of our planet. The microbes that like oxygen and are photosynthetic grow at the top, which is exposed to sunlight and air. The microbes that are anaerobic, which means they don't like oxygen, end up populating the bottom of the column that's devoid of air. Between these two extremes, other types of microbes are distributed according to oxygen levels, pH, and availability of certain nutrients inside the column. The Russian microbiologist Sergei Winogradsky, who is credited as the founder of microbial ecology, invented this method to study how microbial populations naturally segregate in the environment based on their nutritional and environmental needs. The microbes that grow in this column are predictable as they fall within certain metabolic groups. In such a system, the microbes in each layer congregate in the area that provides them with their ideal conditions for growth. The microbial populations that end up in each layer will feed off the waste products from their adjoining layers and provide their own waste that's scavenged by their neighbors too. In this way, each microbial group shares its wealth, as there are always neighboring populations that will use their byproducts as food. You can create this system in a clear glass cylinder like this one, which happens to be 10.5 inches long and about 3 inches in diameter. With this simple yet elegantly contained mini earth, micro whisperers can experiment with different types of light and different types of materials added and be able to observe without a microscope how the microcosm responds by noticing differences in the colors and textures of the strata. This microcosm will give you an intimate understanding of how our land and seas change by the addition of external materials such as pollutants, and shows how the microscopic biota responds to such perturbations.